So now I am going to discuss with you all about the wobble base pairing concept that has been given in your syllabus in your molecular biology. Okay, so let's get started now. So it was first proposed that the specific tRNA anticodon would exist for every codon. Okay, scientists have said, mentioned previously during translation, that for every codon there will be an anticodon sequence, and we all know that anticodon is found within the tRNA. Now if this is the case, then there would be 61 different tRNA, right? Because there are about 64 codons, so and three of them are stop codons, so at least there would be 61 different types of tRNA, right? But experiments were conducted and carried out. Okay, and it was found that by Francis Crick, who also have studied the structure of DNA. Francis Crick in 1966, after finding out the evidence by many scientists, finally Francis Crick have devised the concept called wobble concept. Wobble concept explained these observation that no, there is not 61 different types of tRNA, but obviously the tRNA is carrying the specific amino acids catalyzed by specific amino acyl tRNA synthetase. It normally states that the base which is found at the 5 prime end of anticodon, the base which is found at the 5 prime end of anticodon is not as spatially confined as the other two, allowing them to form hydrogen bond with any of the several bases located at the 3 prime end of the codon. What does it mean? It simply means that let's say for example here, it is saying that the base, the nitrogenous base which is found at the 5 prime end of anticodon, so this is actually the part of tRNA, and tRNA have this anticodon sequence right here, right? Now it is saying that the nitrogenous base which is found at the 5 prime end of anticodon is not spatially confined. That means it's not go for that type of base pairing as we normally know, that adenine will must base pair with here uracil, or it will base pair with then the cytosine, it will be base pairing with guanine, not like that. The base which is found at the 5 prime end of anticodon can bind with several bases found within the mRNA, while the other two nucleotides, they will continue their normal Watson-Crick base pairing. Okay, this is what Francis Crick have proposed. Okay, so I'm again repeating. It means to say that the base which is found at the 5 prime end of tRNA uh, can bind with any of the several bases as proposed by Francis Crick located at the 3 prime end of the codon. So the base which is found at the 5 prime end of anticodon can base pair with any of the bases found on the 3 prime end of a codon sequence in mRNA, while the other two bases will continue their Watson-Crick base pairing concept. Okay, or according to Chargaff's rule you can say. Now not all combinations are possible, with the pairing restricted to those which was proposed by Francis Crick. Francis Crick have proposed some models. I will also show you the table here. Let's continue with the table to clarify this concept. So these are the base pairing combinations according to the Francis Crick, that These are those bases which are found on the extreme 5 prime end of the anticodon sequence of tRNA, and it base pair with the 3 prime end of the mRNA on the codon regions, right? Okay, so it is saying here that the base, the base, if the base is guanine, located at the 5 prime end of the anticodon, it can base pair with either uracil, or it can base pair with either cytosine, found at the 3 prime end of the codon. Now, if the nitrogenous base in the anticodon is cytosine, if the nitrogenous base at the 5 prime end, at the extreme 5 prime end, within the anticodon loop, if the nitrogenous base is cytosine, then it will base pair with guanine on the 3 prime end of your codon sequence in mRNA. If the nitrogenous base at the 5 prime end is adenine, then it can base pair with only uracil. If the nitrogenous base at the 5 prime end is uracil, then it can either bind with adenine or it can either bind with guanine. Maybe the base pairing combination is so much close in case of ribose sugar, due to the ribose sugar, if you compare with the deoxyribose, or if you compare with the normal standard AUGC base pair, here because the ribose sugar, because RNA is made up of ribose sugar, and the distance let them to come closer to interact. Okay, the bases are allowed to interact closer because the ribose distance are very close compared to the normal deoxyribose um, present in the DNA. So that led them to interact actually. If the bases in the anticodon is inosine at the 5 prime in, then it will base pair with either adenine or with uracil or with cytosine. So this was actually proposed by Francis Crick, that this type of base pairing normally occur between the codon and the anticodon sequence within the mRNA, but not all the nucleotides, right? The nucleotide which is located at the extreme 5 prime end of anticodon, they are supposed to be carried out this wobble concept of base pairing. I hope you have understood what I have tried to say. Let us see with the help of image so that you can understand more clearly. See, I have said that if the um, if the anticodon, if the if the nucleotide sequence present at the 5 prime end of the anticodon is uracil, then it can base pair with either with G or with A also, right? So uracil in the first 5 prime end of anticodon can base pair with either with adenine or guanine. Similarly, in the other structure, you can see that if the in the other structure you can see that if the base found at the 5 prime end is inosine, okay, it can base pair with either with uracil, or it can base pair with cytosine, or it can base pair with adenine as well. Well, inosine is actually the fifth base which is found in the anticodon sequence of tRNA. Okay, it is the fifth nitrogenous base which is found within the anticodon sequence of tRNA, and inosine occurs through the result of enzymatic modification of adenine, whose carbon number 6 is deaminated to give rise to the 6 keto group of inosine, right? So due to deamination of adenine, it gives rise to inosine, and this inosine found at the 5 prime end of anticodon on each tRNA can able to base pair with uracil, it can base pair with cytosine, or it can base pair with adenine in the mRNA, in a codon sequence of mRNA. Okay. So Francis Crick have devised this wobble concept to explain these observations, and he have stated that the nitrogenous base, which is found at the 5' end of anticodon, is not spatially confined. That means it does not follow that Watson-Crick rule of base pairing. It can form hydrogen bond with any of these several base which is located at the 3' end of the codon. And it is not like as you like you can combine with any of the bases, but not all combinations are possible. That's why I have shown you the table which was proposed by Francis Crick, that this type of combinations can come. For example, uracil at the wobble position. That so this position is called wobble position. The position at the 5' end of the anticodon is the wobble position. Okay, so the position found at the 5' end of the anticodon is the wobble position, because it has a variation in base pairing with the codon sequence. So, like suppose uracil, as shown in the picture, at the wobble position can base pair with either with adenine or it can base pair with guanine, as we have seen in the table in the fourth row, that uracil found in the anticodon at the 5' end can base pair with adenine or it can base pair with guanine on the codon sequence in mRNA. Okay. While inosine, if it is present in these same wobble position, at the same wobble position, that means at the 5' end of the anticodon, if there is inosine present, then it can base pair with either with uracil or with cytosine or with adenine. And these pairings permitted by the wobble rules are those that, given ribose distance are so close, because these whole are RNA actually, right? All are RNA. This one is tRNA and this is mRNA. All are RNA, and RNA is made up of ribose sugar. Because the distance, because when base pairing occurs, the distance between the ribose sugar is not too much far away, very close. That's why this type of unusual base pairing, wobble base pairing, comes to be seen. Okay, purine with the exception of inosine adenine pair or pyrimidine pairs would give rise ribose distance that are too long or too short respectively. So this is what wobble base pairing actually occurs. Now, however, wobble rules do not permit any single tRNA molecule to recognize four different codons. The wobble rules do not permit any single tRNA molecule to recognize more than four codons. These codons can be recognized only when inosine copies the first 5' position of the anticodon. So three codons can be recognized. Okay. So not more than four codons can be recognized by any single tRNA. Like for example, to support this wobble concept, one example is I am showing here with you. Like at least at least three tRNA will be existing for six serine codons. The six serine codons, that means the codons which will code for serine are what? UCU, UCC, UCA, UCG, AGU, and AGC. So these are the six different codons which code for serine amino acid. And for this we require at least 3 tRNA which will recognize these six codons in the mRNA. The other two amino acids, leucine and arginine, that are coded by 6 codons also have different tRNA for the set of codons that differ in the first position or second position. So according to this concept, the wobble rules does not permit any single tRNA molecule to recognize 4 different codons. So here you can see for 6 codons, at least 3 tRNA molecules will be needed to recognize all of them. Not more than 4. Okay, not 4 codons will not recognize by a single tRNA. That should be that has been mentioned in the wobble hypothesis. Then the other two amino acids like leucine and isoleucine, sorry, leucine and arginine. Now for leucine and arginine, if I say for leucine and arginine, leucine is coded by CUU, CUC, CUA, CUG, and arginine, right? UUA, UUG. Okay, so we see 1, 2, 3, 4, 5, 6. We get 6 here. Okay, leucine is coded by 6 uh, codons, and arginine is also coded by 6 codons. Arginine is also coded by 6 codons. We have to see. Okay, CGU, CGC, CGA, CGG, AGA, and AGG. These are coded by arginine. So all these 6 codons will not be identified by a single tRNA. Obviously, it has been mentioned that multiple tRNA will be involved for recognizing these 6 codons. But for each codon, there is not a single tRNA that we have seen in this wobble hypothesis. That one tRNA can recognize more than one codons as well, coding for the same amino acid. Okay, so this is what about the wobble hypothesis is actually. Now, if you see the three-dimensional structure of tRNA, the three anticodon bases as well as the two following 3' bases in the anticodon loop all pointed roughly in the same direction with exact conformations, largely determined by interactions between the flat surfaces on the bases. Uh, I think it has been given in the figure. No, it has not been given in the figure. So it has been mentioned here that if I draw, then I can able to explain with you. So it has been mentioned here that the in this three-dimensional structure of tRNA, the three anticodon bases as well as the two following 3' bases in the anticodon loop all roughly pointed in the same direction with exact conformations largely determined by stacking interactions between the flat surfaces of the bases. So if you see the three-dimensional structure of tRNA, it somewhat look like this. It somewhat look like this. Okay, this is your 3' acceptor arm, this is a 5' acceptor arm, and suppose this is your anticodon loop, anticodon arm and anticodon loop, and here is your where anticodon sequence is present. Okay, this is the D loop, D loop. This is the T loop. Okay, this is the acceptor arm. Acceptor arm. Okay, this is the D stem. So it is saying that here, according to the notes that I have given to you, that the three anticodon bases as well as the following three prime bases in the anticodon loop all pointed in the same direction. So they are all pointed towards the same direction, right? Now, um, exact conformations largely determined by interactions between the flat surface of the bases. So the first 5 prime anticodon bases is at the end of the stack and is perhaps less restricted in this movement than the other two anticodon bases. Hence, oval in the third 3 prime position of the codon. Okay, so here it is saying that all are pointing towards the same direction, and here it is saying that the 5 prime anticodon basis is at the end of the stack and is less restricted in its movement. It is not like that, it is less restricted, so it can able to base pair with the other bases in the codon sequence in the at the 3 prime position of the mRNA. By contrast, the other two bases are appear to be bulky, modified, and thus restriction of its movement may explain why wobble is not seen in the 5 prime position of the code. So it is just a conformational type that wobble base occur only on the 5 prime position. Okay, so here we can see that the 5 prime position anticodon sequence is perhaps less restricted in its movement compared to the other two anticodon base, and that's why they have this wobble base pairing concept can be explained from this model, from this structure, from by studying this model. Okay, so this is the whole notes regarding the wobble hypothesis. Just go through these notes, and for any problem you can ask me in the WhatsApp group. Okay.